Today we get to talk about plant growth. And before we get into all of the details and the mechanisms that enable plants to increase in size, let's talk about one interesting fact regarding plant growth. See, plants can continue growing for as long as they live and conditions in the environment are favorable. This is something we call indeterminate growth. Think, for example, about a shrub or a tree. Those types of plants that live year after year after year can continue growing upwards, increasing the amount of branches and stems that they have, and also downwards in the direction of more water, and that is going to be what roots are going to be doing. See, in contrast to animals that actually have limits to what is the size, the maximum size, or how big they can get, plants can continue growing indefinitely. So the growth in animals is called determinate growth, and it's going to be determined by the limits set in their DNA. Now, there are going to be some parts of a plant that will not continue to grow indefinitely, and that is going to be organs like leaves, flowers, and fruits. They reach a maximum size, and then they don't grow anymore. What enables a plant to grow is going to be specialized tissues called meristems. And these meristems are going to be located in strategic places in the plant, like you see in this illustration that I'm using for this presentation. At the tip of a plant, there is going to be a shoot, which is going to consist of a stem and leaves. So right there, the very tip of the shoot, there is going to be an apical meristem. And also down here, where the roots are growing, there are going to be also apical meristems. Those apical meristems are going to be responsible for a type of growth in plants we call primary growth. Primary growth should be easy to remember. That is going to be the growth a plant does vertically, on a vertical axis. And so that's going to be growth upwards towards the sun and downwards in the direction of water. And this is going to be accomplished by what we call apical meristems. And so right there, as I mentioned before, at the tip of the shoot and at the tip of the roots is going to be where these apical meristems are going to be located. Secondary growth is going to be different. Secondary growth is going to be accomplished with the help of lateral meristems. And here I want you to notice something interesting too. See, for primary growth, the meristems at the top and the bottom of the plant, at the shoot, and at the root tips, we just call them apical meristems. But when we look at lateral meristems, there's actually going to be two types. And one is going to be known as Vascular Cambium, which is responsible for adding new layers of vascular tissues. We now are familiar with the names of those tissues. We call them xylem and phloem. Now, there's also going to be another type of lateral meristem, and that is going to be called Cork Cambium. And this is going to be replacing the epidermis of the plant and replacing it with something, a layer called the periderm. Peri means like the perimeter, the outer perimeter of the plant, and that derm is going to be making reference to a protective layer. The periderm is going to be, of course, thicker and tougher than the epidermis a plant had when it was just beginning to grow, as it had just emerged from the seed through the process of germination. What I wanted to point out to you here with regards to these lateral meristems, and this is just as an introduction, we will be exploring in greater detail secondary growth on a separate video that I'm going to make. Take a look at this vascular cambium. The vascular cambium can be seen here as a red line. It's actually more like a cylinder, and it's going to be only one layer of cells in terms of thickness. Towards the core of the plant, there's going to be new layers of xylem that are going to be added, and towards the periderm, there's going to be new layers of phloem. And as I said, those details, we're going to be exploring them in greater detail on a separate video. Here we can see also an illustration of the Cork Cambium. And this Cork Cambium usually originates from the outermost layer of cortex. And that is going to be a layer of cells that is going to be producing additional cells that are going to be known as cork. What people typically call the bark is actually, in botany, is often referred to as cork. Relevant also to the growth of plants is going to be the concept of the longevity and the life cycle length of plants. There are going to be many plants that, for example, are going to be completing the entire life cycle from the moment they germinate from a seed to the moment they grow and flower and produce seeds. All of that is going to be completed for many plants within 1 year or less than a year. These kinds of plants, we're going to call them annuals. There are going to be plants that actually require 2 years to complete their life cycle, and we're going to be calling them biennials. The bi means 2 in this case. Annual makes reference to year. And there are going to be many plants that are going to be requiring many years to complete their life cycles, and we're going to be calling those plants perennials. In this illustration that I have for a plant that happens to be a perennial where many years of growth, you can appreciate other details regarding the anatomy of a plant. And so you can see here that from a node, which is going to be the place where one of the axillary buds, it's going to be on one of the primary meristems, used to be a section of the plant, a new shoot grew, and there is going to be another place where a new year's worth of growth is going to be beginning. So from node to node is going to be a region we call an internode. And this is going to be corresponding to what can grow, a plant can grow within a year. So you can see this bracket here, 1 year old side branch that is going to be originating from this axillary bud. There are also going to be bud scars on a stem. And there used to be maybe a whorl, like a circular arrangement of buds that existed the year before. And you can see here now that there's going to be another growth that is going to be going more on a vertical axial type of arrangement. This is going to be lateral growth, and this is going to be the primary growth that is growing more in terms of height, in terms of plants' vertical growth. There's also going to be other new buds that may at any moment on future years lead to additional growth, and that is going to be typical of plants that have many years worth of growth. What I want to emphasize in this video is more of the details about primary growth. Let's focus on the root. And I like this illustration that I'm showing right now because we can see some of the details of the root that is going to be growing in size. This region you see over here, I hope you can see my pointer, which looks like a hand, is going to be the zone, the beginning of the zone of cell division. So there is going to be an apical meristem right here that is going to be producing cells on this side. Those cells are going to be becoming part of the cortex, which surrounds the vascular cylinder. Some of these cells are also going to be giving rise to the cells of the vascular cylinder, which will include the xylem and the phloem. Other cells that are originating from this apical meristem are going to be forming cells that provide a protection for the root. See, as the root is growing in depth, sometimes may encounter a stone or other things that may harm this region or zone of division. And this is a sensitive region. The plant wants to protect it. And that protection will be provided by cells of the root cap. So in a direction away from the older part of the root, there's going to be cells that are forming the root cap. And in the direction of the older part of the root, there's going to be new cells. Some are becoming part of the cortex, and some are going to be becoming part of the vascular cylinder. Here, there's going to be other zones we can recognize. So in addition to the zone of cell division, these cells, as we go up to older parts of the root, are going to be the cells of the zone of elongation. So these cells here are getting longer. The older they get, the longer they get. Again, some of these cells are going to have different functions. Here we're looking at this yellow section, which is going to be part of the cortex. It's going to be a type of ground tissue. And here in purple, we're going to be seeing the cells of the vascular cylinder of a root. As we continue to go higher up, we're going to be finding the zone of differentiation, where cells have by now been around long enough that they have acquired their own mission in the life of the plant. So these cells, for sure, now can be used, for example, for the storage of nutrients. These cells of the vascular cylinder are going to be participating in the transportation of materials, and the cells of the epidermis are going to be, for sure, providing protection to the root. So that is with regards to roots. The primary growth of roots is going to be responsible for producing everything: epidermis, ground tissue, like this cortex you see here, which is going to be a type of parenchyma. Here we're going to see the vascular cylinder, which is going to be characterized by, in the case of eudicots, this X. These are going to be cells that correspond to the xylem. These larger in diameter are usually going to be vessel elements. These are going to be smaller in diameter, usually going to be tracheids. Here we see the cells of the phloem. These are usually going to be sieve tube elements. This outermost layer of cells that are part of the vascular cylinder are going to be forming something called the Peri Cycle. And this Peri Cycle is going to be important because, as we're about to see, secondary roots will begin to form from this layer of cells, from this Peri Cycle we are talking about right now. The endodermis is going to be an important tissue also. We're going to be learning more about the function of the endodermis when we get into the next chapter that deals with transport of materials within plants. This is going to be like a barrier that selects what substances, water, and types of minerals that can be going into the plant, into the xylem, to be transported upwards towards the top of the plant. In many monocots, there's going to be a core of parenchyma cells. You can see that core of parenchyma cells right here. This is the type of ground tissue. And this is going to be surrounded by cells of the xylem. Remember, always the largest in diameter. These are going to be most likely vessel elements. And there's going to be maybe some tracheids over here. In addition to that, there's going to be phloem. The phloem is going to be constituted by cells known as sieve tube elements. Now here we can see a ring. This is a black line that was drawn by the artists illustrating our textbook. This outermost layer of the vascular cylinder is going to be the Peri Cycle, and out here, which is darkly stained red, that is going to be the endodermis. This is going to be the layer I told you just a moment ago will help regulate the materials in solution that will be allowed to pass into this vascular cylinder to be transported upwards towards the top of the plant. Not everything that is absorbed by the root will be transported within the plant. And this is going to be that selective barrier. These cells here are going to be doing that, deciding what continues to move and what doesn't. All right. So ground tissue in a root is going to be mostly parenchyma cells. You may remember from class material that these parenchyma cells are going to have metabolic functions. In this case, the darkly stained nature of the cortex, the parenchyma we're dealing with here, is going to be making complex carbohydrates like starch. And so that is what you see there, the little starch granules that have been stored for the plant to use as a resource, as a root may need that resource to continue growing deeper into the soil. Here we see again that layer we called before, the epidermis on this slide. I'm sorry, the endodermis. Here on this slide, you can see it labeled, and you can see that this endodermis is going to be the innermost layer of the cortex of this parenchyma type of tissue. And that is where a barrier will exist to prevent materials unwanted by the plant from continuing to move into the xylem. Lateral roots are going to be emerging from the outermost layer of the vascular cylinder, that is the periderm. So from here, from the periderm, lateral roots will be emerging. That is convenient because there must be a connection between the transport of nutrients from the center of the vascular cylinder, where the xylem is going to be, providing water for these lateral roots. Additionally, materials like sugars can be transported from the phloem over to this growing and developing lateral root. Shoot growth is going to be pretty much like I said before. Here we see an apical meristem. Cells are going to be added in this direction, and cells are going to go through a process of maturing and becoming eventually differentiated, adopting a function. Here, some of these cells are going to become part of the cortex. Some cells are going to be forming the primary phloem and the primary xylem, the very first vascular tissues the plant is going to use for delivering materials up here to the top of the plant, where the apical meristem is going to be. And that's going to be all that I'm going to say with regards to the primary growth. Watch for another video where I will be discussing secondary growth.